well, nā mai haere mai, tēnā koutou, tēnā koutou, tēnā koutou katoa. So very, very warm welcome to everyone for today's Genomics Aotearoa seminar. We're really delighted to be hosting Dr. Emily Koot. So Genomics Aotearoa, for those of you who don't know, was established in 2017 as a collaborative platform to facilitate and promote genomics research within Aotearoa. And our Genomics Aotearoa whānau includes researchers and collaborations from 9 partners, including 4 Crown Research Institutes and 5 universities. So Emily and myself are part of a Genomics Aotearoa project with a bit of a mouthful of a name. It's called High Quality Genomes and Population Genomics. And we do all sorts of fun genomics things across both environment and primary production spaces. So as part of that project together, it's my real pleasure to introduce you today to Dr. Emily Koot. So Emily completed both her honours and PhD studies based with the fabulous Professor Mary Morgan Richards and Professor Steve Trewick at Massey University before taking a position around 4 years ago as a scientist at Plant and Food Research based in Palmerston North. So Emily uses genomic datasets and tools to explore the genetic diversity, population dynamics and landscape genomics of plant and animal species, and that's included already to date manuka, rewa, kiwi fruit, hoki, apple, kauri and kakapo. And she also continues to work on research that emerged from her PhD, looking into the effect of climate change on New Zealand's alpine ecosystems. So Emily tells me that she enjoys the versatility of population genomics and all of the different applications it has across society and industry. So I believe Emily is going to dive into today one of the many projects that she's involved in and tell us about how genome-wide analyses have revealed the genetic stock structure of Hoki. So thank you very much, Emily, for accepting the invitation, and please take it away.
Okay, kia ora koutou, ngā mihi mahana, ko tararoa ta maunga, ko manawatu te awa, no te papioia oho, ko Emily Koot. Hi everyone, and thank you all for coming along to hear me speak today. Today I'm going to talk to you about a project we completed last year where we investigated the genetic diversity and population structure of Hoki, Macruronus, novaezelandiae. So here's a quick overview of my talk today. First, I'll introduce our team before giving a brief overview of fisheries genomics being applied around the world. I'll then introduce our study species and our project objectives before moving through the methods and results of the study and what we plan to do next, and then I'll wrap things up. So here's our team, as I'm only one of many people who have contributed to this work. I'll introduce myself on the next slide, but then next there's Chen. Chen's a research technologist in bioinformatics at Plant and Food Research based in Auckland. Chen's areas of expertise include genome and pangenome construction, variant discovery, gene expression profiling, and polyploid genomics. Then there's Igor, and he's a research associate at Plant and Food Research based in Nelson, and he manages the fish physiology and molecular biology labs for our seafood production team. Then there's Elena, also a scientist at Plant and Food based in Auckland, whom many of you will know as an absolute magician and fountain of knowledge when it comes to high quality DNA extractions and preps. And then we've got Roy, and Roy's a bioinformatician at Plant and Food based in Te Puke, and he's an expert in transcriptomics, high throughput computing, and general data wrangling. And then we've got Richard. Richard's a Richard's a fisheries expert based at Deepwater Group in Auckland. The Deepwater Group is a non-profit organisation working to ensure New Zealand's fisheries resources are managed to optimise their long-term sustainability. And then we've got David and Maran, who I'm sure most of you already know, both science group leaders at Plant and Food Research based in Palmerston North and Nelson, respectively. So David specialises in applying genetic mapping and genomics to identify genetic control of plant characters, and Maran is an expert in evolutionary biology and ecology, applying genomic tools to better understand adaptive and non-adaptive evolutionary processes. So that a little bit about myself. So I studied at Massey here in the glorious Palmerston North for 8.5 years, completing my PhD in 2018. My honours project looked at the diversity and diversity of Purputus, or velvet worms as you may know them, and my PhD looked at the effect of climate change on the diversity and distribution of New Zealand's endemic alpine grasshoppers. So my early research predominantly focused on invertebrates, and I still have quite a soft spot for insects. And for the most part, my research, the research I've been involved in revolves around evolutionary biology and ecology, such as population genomics, landscape genomics, phylogeography, and ecological niche modeling. So after my PhD, I picked up a job as maternity leave cover in David's team at Plant and Food. And fortunately for me, they decided to keep me on, and I've now been there for nearly 4 years. It was when I started in this team that I shifted into working with genomic scale datasets. And it was a bit of a shock to the system, moving from compact, small mitochondrial genomes to working with millions of variants. And it's been a massive learning curve and challenging, but it's been really satisfying, and I've learned heaps. In my current position, I use genomic datasets and tools to explore the genetic diversity, population dynamics, and landscape genomics of lots of different plant and animal species, as Anna mentioned. And at Plant and Food, the field of population genomics is steadily expanding, and we've got a number of projects on the go, such as we've been involved in the NBIE-funded Honey Landscape project, looking at the genetic diversity and structure of Manuka and Rewa in order to help Maori agribusinesses understand their genetic resources. I'm leading a research project looking into the genetic diversity and structure of our kiwi fruit germplasm collection, which is held by Plant and Food, to help inform breeding programs. We've developed a multi-species snip array, which will aid in genotyping, breeding, and screening some aquaculture species, berries, and Manuka. I'm involved in the Genomics Aotearoa Kakapo 1 to 5+ project. And then, of course, the reason for today's talk, we've been applying population genomics tools to better understand fishery species in New Zealand. So which brings me to today's topic. Before I get into the specifics of our project, I thought we could take a step back and have a quick look into the role of genomics in fisheries management. The effective and sustainable management of fisheries is vital in order to avoid overexploitation of local populations and entire species, and the management of these fisheries has major downstream effects on wider ecosystems, industries, and of course the people and communities that depend on them. So for effective fisheries management, it's important to know the genetic structure of species, the size and range of populations, their genetic diversity, and the level of connectivity between these populations. Now, in the past, it's not always been easy to achieve this using traditional means. Populations can be expansive and difficult to quantify, and political boundaries and management units may limit sampling and not reflect the true state of wider populations. And phenotypic differences between populations may not always reflect actual genetic stock structure. Smaller scale genetic approaches such as enzymes, microsatellites, and mitochondrial DNA sequence data have not always had the power to reveal cryptic stock structure. And this is where next generation sequencing and genomics has become a really useful tool in the fisheries management toolbox. So where traditional genomic methods may have only considered a small number of often neutral genetic variants, genomic approaches allow us to look at variants across the entire genome, numbering in the tens of thousands to millions, and greatly improving the statistical power and reliability of the results. So now neutral, adaptive, and structural variants can all be explored to answer pressing questions relating to states of fisheries stocks. And like for so many other areas of research and industry, genomics is becoming so much more affordable and making it more feasible to apply. And the more genomic resources that are existing out there, the easier it's becoming to integrate it into management strategies, as it's making it faster to assemble and annotate genomes. Genomic approaches can also be used to do things such as age fish, explore adaptive capacity of populations to things such as climate change, and understand diet, monitor ecosystems, examine taxonomy, and can also be used for traceability purposes. I thought I would just quickly mention some very general life history traits that characterize marine fish species, as these are quite important things to consider. So marine species tend to have very large populations that can cover vast geographic areas. Plankton and larvae can be carried very long distances, sometimes hundreds of kilometers from where they hatch. As adults, they can also be highly highly migratory, traveling long distance between spawning and feeding grounds, and they typically have few barriers to gene flow. So this means that populations tend to be highly connected, and this can make it really challenging to characterize fine population structure. Often fishery species are reported as having shallow population structure, but this is usually based on a small genetic investigation using neutral loci. And it's often been assumed that because of the high levels of gene flow that fisheries populations experience, it has a homogenizing effect on allelic frequencies between populations, limiting the potential for local adaptation and thus limiting structure to form. But genome-wide studies all around the world are challenging this view. So it's now considered that due to the very large effective population sizes of many fishery species, genetic drift has little effect, and locally beneficial alleles can get the chance to sweep through populations. And now there are lots of really cool, interesting studies emerging in fisheries genomics area covering a wide range of taxa, many of which reveal a mismatch between managed units and actual genetic stock structure. And these studies will likely, hopefully, have implications for how these fisheries will be managed going forward. And so, yeah, these studies emphasize the importance of examining variants across the entire genome and investigating outlier Snips when trying to determine the genetic stock structure of fishery species. So when it comes to fisheries genomics in New Zealand, it's an emerging field. I've just got some quick stats here for you about New Zealand fisheries. So New Zealand's EEZ covers some 4.1 million square kilometres, as shown here on the map, and that makes it the 6th largest EEZ in the world. And over 15,000 marine species inhabit that area, and although lots of those are migratory, a lot of them are unique to New Zealand. The deepwater fisheries industry alone contributes some $1.8 billion to the New Zealand economy, and approximately 600,000 tonnes are harvested annually from wild fisheries and aquaculture around New Zealand, and this comprises over 100 different managed species. In a recent review, they found that genetic resources existed for 23 out of 86 of those managed fish species. But when it comes to genomic resources, these only exist for a few species to date. And this includes the species that I'm going to talk to you about today. So Hoki, also known by other names such as blue granaria, is a deep water teleus species. It's slender and silvery in appearance, and they range from 60 to 100 centimeters in length and can weigh up to 3.5 kgs. They're abundant between 200 and 600 meters, and feed on squid, crustaceans, and other fish. They become reproductively active anywhere from 3 to 7 years of age, and they can live for up to 25 years. So Hoki is found in the southern hemisphere and is distributed around New Zealand, Australia, the subantarctic islands, and around the southern coast of South America. So this map shows roughly where they're likely to be found, and you can see that they're quite concentrated around the coastline of New Zealand, and especially out to the Chathams and down to the subantarctics there. So Hoki is considered a taonga species by Maori, and it's an important source of kaimoana, and a number of iwi have Hoki quotas. Hoki is New Zealand's most abundant fishery species, and it's been the largest commercial fishery in New Zealand since the 80s. And since 2001, it's had Marine Stewardship Council Fisheries certification as a sustainable fishery, and this certification has been successfully renewed for 4 consecutive times. So Hoki has a highly migratory and complex life history. For fisheries purposes, Hoki is currently managed as 2 separate stocks, a western stock and an eastern stock. So just going to explain this in a bit more detail. So the western stock is thought to spawn on the west coast of the South Island. Larvae and juveniles are then transported to the east coast and Chatham Rise nursery and feeding grounds via the Westland and D'Urville currents. And then at about 4 to 6 years in age, the young adults then migrate down to the subantarctic waters, and this is considered to be their home feeding grounds. Mature adults will then migrate between here and the spawning grounds of the west coast. For the eastern stock, they're thought to spawn in the Cook Strait, as well as along the east coast of the South Island. Larvae and juveniles are then transported to the Chatham Rise nursery and feeding grounds, and here they mix with the eastern stock larvae and juveniles. Eastern stock fish then remain in this area as young adults, and mature adults will move between the Chatham Rise and Cook Strait spawning grounds. So the two-stock model was developed based on surveys that found fish in different regions mature at different rates and vary morphologically. And the two-stock model assumes that adults from the two stocks remain segregated during spawning, which maintains this two-stock structure. Now, there have been a couple of small genetic studies in the 80s and 90s using cytochrome B sequences, RFLPs, and microsatellites, but none of these were able to identify any significant genetic structure. And no genomic data has been used to help inform this model or management of the species. Which brings me to the aims of this project. So there's still a lot we don't know about this species. For example, we don't know how Hoki is genetically structured, how many subpopulations there are, if there are subpopulations, how genetically distinct they are. We don't know how genetically diverse the species is across its local range, and we don't know for sure whether or not adults segregate, as hypothesized by the current two-stock model. And these are all questions that Deepwater Group was really interested in answering. So in collaboration with Deepwater Group, Te Ahu Kaimoana and Kaitiaki, we set out to try and answer some of these questions and help with the management of the species. And to do this, we proposed to carry out a comprehensive genomic analysis to investigate the genomic stock structure of Hoki by first assembling the genome, and then generating a catalogue of genome-wide Snips to carry out population genomic analyses. And then thirdly, we identified regions of the genome under selection. First step, of course, was to engage with Te Ahu Kaimoana and Kaitiaki before any operational aspects of the project were initiated, and to gain informed consent before commencing the study. As mentioned, Hoki are widespread and highly migratory around New Zealand, and they are therefore of interest to many iwi and hapu around the country. Te Ahu Kaimoana represents 58 iwi who hold fishing quota, and they were able to facilitate discussions with everyone who was interested. And a working group was established to advise on the suitable sampling protocols and the management and storage of samples and data before, during, and after the project. And this included an agreement to store the genomic data raw and analysed in the Aotearoa Genomics data repository. So in May 2020, a female Hoki was sampled from Cook Strait for the Genome Assembly, and following DNA extraction by Elena, this was sent to AGRF for short and long read sequencing. The Illumina NovaSeq 6000 was used for the short read sequencing, and for long read, they used Oxford nanopore technology. Also, between May and September 2020, fish were sampled for the population genomic analysis. For this part of the study, we used MPI fisheries observers who took fin clips of fish caught on commercial fishing vessels. The observers also collected data on fish size and sex alongside the GPS location of each trawl. We wanted to make sure we collected fish at different life stages so that we could investigate the two-stock model. So we sampled juveniles, adults, and spawning fish. So fish were sampled from 12 sites around New Zealand and 2 sites in Tasmania, which you can see on the map there. The known spawning grounds that were sampled are here on the West Coast, number 3 and 4, and here in the Cook Strait, number 5 and 6. So the fin clips were stored in 95% ethanol, and 30 to 40 samples of each site were sent to Neogen GeneSeq for DNA extraction and whole genome resequencing. So in total, 510 Hoki were sequenced from 14 locations for the population genomic part of the study. Chinwe from Plant and Food carried out the Genome Assembly. And this is the first high quality genome assembly for Hoki, which is a great resource to have, and of course necessary for the genome-wide variant calling. So 22.7 GB of sequencing data was generated for the assembly with a N50 of about 5 KB. Chen used Fly to assemble the genome, which totaled 501 megabases in length and comprised 566 contigs. The longest scaffold was about 26 megabases in length. So a BUSCO analysis using the conserved actinopteri GI gene set gave a score of 93.8%, so which is awesome and really it's highly comparable with other recently assembled fish genomes, such as the Atlantic cod and Chinook salmon. And the genomes in the process been assembled into 21 chromosomes. For the population genomic analyses, just over 6,000 GB of Illumina data was generated, which worked out at about 12 GB per sample at a mean depth of 24x, based on the genome that was assembled. And even individuals with the lowest yield still had greater than 10x coverage. The median mapping rate to the reference genome was 93%, and in total 3.9 million variants were detected using Freebase. After filtering this for Snips and VSCF tools, applying these parameters here, we were left with 227,490 Snips to carry out the population genomic analyses with. After SNP filtering, we wanted to detect SNPs under selection, so we could carry out the analysis on these outlier SNPs, neutral SNPs, and all SNPs combined. To do this, we applied 3 different methods: Test3, PC-Adapt, and Leah. Each of these methods searches for variants under selection in slightly different ways. Just in brief, Test3 applies matrix factorization algorithms to allele frequencies. PC-Adapt runs genome-wide selection scans based on principal component analyses, and Leah calculates FSD statistics from ancestral allele frequencies. The 3 analyses were carried out on 2 different data sets. The first contained all filtered SNPs and all Hoki samples, so all 510 samples, and the second set contained all filtered SNPs, but only New Zealand samples. So that was 450 samples. The R package Q value was used to test for statistical significance, and we applied a FDR threshold of 0.05. And variants that met this threshold for all 3 analyses were considered as outliers that were putatively under divergent selection, whilst any variant that was not considered under selection by any of the analyses was considered to be putatively neutral. And so subsequently, we had 6 SNP data sets to carry out analyses on. So these are the analyses and tools we use to carry out the population genomic part of the study. Summary statistics were calculated in VCF tools. Pairwise FSD distances were estimated using the R package stamp. K-means clustering and discriminant analysis of principal components was carried out using ADGenet. Ancestral admixture was examined using Leah, and effective population size was explored using SNP and any estimator. Okay, on to some results. So we produced lots of results for this project. I'll just cut to the chase and go through the most important findings and try and keep things simple. First, I'll go through some of the results for the all SNPs, all samples dataset. So we carried out the K-means clustering analysis on the dataset, and it identified K to be 2, which is the top left graph there. When we examined these clusters further via DAPC analysis, the 2 clusters clearly identified 2 geographically and genetically distinct groups, one containing all New Zealand Hoki samples, which is the cluster on the left there, and one containing all the Australian Hoki samples, so the cluster on the right. Similarly, LEA, the Leah ancestral admixture analysis also identified the same 2 clusters, and it's shown in as demonstrated in the bottom plot there. When we looked at the genetic differentiation within and between the 2 clusters, there was little genetic differentiation within the New Zealand samples or within the Australian samples. But we did find FST to be much greater between New Zealand and Australian samples, although still fairly low at 0.0214 there. And these same analyses were carried out on the neutral SNP data set with all Hoki samples, but the findings were they're very similar. So we wanted to be certain we weren't missing any substructuring within the New Zealand samples. So we carried out analyses on just the New Zealand samples as well. And here I'm presenting the results for the neutral SNP data set, but just the New Zealand samples. So K-means clustering identified just 1 cluster within the data set. And you can see the results of the DAPC analysis there on top right. So lots of overlap between sampling sites. And you can see 2 sites appearing to spread out a bit, FMA3J1 and FMA6 SNERs. But I'll come back to that. Similarly, the Leah ancestral admixture analysis identified 1 cluster, and you can see a similar pattern there with those same 2 sites. So mean FST within New Zealand populations was again really low. And although the heat map displayed here appears to be the 2 populations, FMA3J1 and FMA6, appear to be slightly distant from other populations. When we look at the population replicates, so FMA3 or other FMA3 samples, FMA6, Norgay, they're all sitting with all the other populations. So it appears to be some kind of weird, spurious thing going on there. And we think it may have to do with sequencing quality rather than any actual hidden structure. But that's something I still need to dig into. And then, so we carried out these same analyses on the, with all SNPs and just New Zealand samples. And again, the findings were near identical. So this brings us to the data sets consisting of adaptive SNPs only. So our outlier scans identified a number of SNPs under selection. For the data set consisting of all Hoki samples, Test 3 identified over 8,000 SNPs, PC Adapt identified 3,500 SNPs, and Leah identified nearly 10,000 SNPs. And 2,797 of those met criteria across all 3 analyses and retained for downstream analysis. And when we carried out the scans with just the New Zealand samples, Test 3 identified nearly 3,000 SNPs, PC Adapt identified 3,700 SNPs, and Leah identified over 5,000 SNPs. So, but only 10 of those we identified as beautifully under selection across all 3 of the analyses. So here I'm displaying results for the New Zealand only data set. And we weren't able to identify any structure within the 10 adaptive SNP data set. And the FST values also remained really low between populations. The adaptive SNP data set, including Australian samples, displayed a similar pattern as we'd seen earlier, separating New Zealand and Australian samples into 2 distinct clusters. But with that data, those results, the pairwise FST distances between New Zealand and Australia were much higher at 0.32. So we detected over 2,700 adaptive SNPs when the Australian samples were included, but only 10 within the New Zealand dataset. And that indicates that the Australian Hoki may have a higher adaptive potential than the New Zealand Hoki, but further analysis is required to better understand the adaptive variants. We also carried out analyses to investigate the effective population size of New Zealand Hoki. So using all SNPs and just New Zealand samples, we thinned the data set by 10,000 sites, and that left us with a SNP data set just under 3,000 SNPs for analysis, and we carried out a contemporary LD-NE analysis using NE estimator, and this estimated NE for NZ Hoki to be about 133,000. However, the confidence interval was huge, ranging from 39,000 to infinity, so not very reliable results there. We also looked into historical NE using Snips, and this estimated NE to be about 229,000, nearly 1,000 years ago, generations ago, sorry. And NE has gradually declined to 43,000 in 189 generations ago. So the main findings of the study, we found clear genetic differentiation between New Zealand and Australian samples, and that was found across neutral and adaptive loci. We found weak genetic differentiation within the New Zealand samples, and no evidence of substructuring or evidence of two genetically distinct stocks. So, you know, this indicates there's a mismatch between currently managed units and actual genetic units. And although I didn't show the results for it, we did look into whether there was any genetic differentiation evident in samples of different life stages, but we found no evidence of substructuring within our juvenile, adult, or spawning samples. We were able to identify a number of outlier SNPs, which indicate that the fish are undergoing local selection and that adaptive SNPs can accumulate, you know, within these massive, highly connected populations. And yeah, our effective population size analysis indicates a decline in Hoki population size in New Zealand over time, but yeah, it's quite hard to get reliable results for NE. So we encountered a few challenges during this project, and yeah, the key one being, you know, yeah, getting a reliable effective population size estimation. To get accurate results, you need to sample at least 1% of the population, and that's just not really feasible or affordable in this situation. Close-kin mark capture looks promising, but again, likely too expensive to apply here. And it would have been really interesting to get an idea of how genetic diversity and structure of Hoki has changed over time, especially once it became managed, but yeah, we just don't have the data for that, unfortunately. And then there was a whole different kind of challenge where couriers destroyed 4 boxes of samples. So that, you know, that happened as well, but we were able to still get heaps of samples anyway, but that was a thing. So what next? Yeah, our paper got published, so that was awesome. We have some additional samples to add to the data set from northeastern New Zealand and subantarctic/Antarctic area. We have the sequence data back for these, but yeah, I think variant calling is underway, and then yeah, we need to combine those data sets. The genome has been assembled into 21 chromosomes, as I mentioned, and then once that's done, we can build a linkage map. We also plan to look into structural variants and see if that reveals any structure that we weren't able to find in this study. And I've made a start on carrying out gene environment association study. And I've got some preliminary results to share with you today. So just a quick run through of what I've been working on. So this is for just the New Zealand samples. I downloaded environmental data from Bioracle, which is a database specializing in bioclimatic layers for global marine environments. For our analysis, we chose 12 variables of interest, and then we used the latitude and longitude information of each Hoki sampling site to extract data, environmental data from these layers, and use PCA plots and pairwise Pearson's correlation coefficients to check for collinearity between variables. And after checking for that, we narrowed it down from 12 variables to 7 of that we consider to be ecologically relevant and of interest. So those were mean current velocity, mean dissolved oxygen, mean nitrate, mean phytoplankton, mean salinity, mean temperature, and primary productivity. And so they used LFMM2 and BayPass to carry out the GEAs, and outputs were filtered by FGR threshold of 0.05. The two datasets were compared, and Snips occurring in both datasets were considered to be putatively associated. And yeah, so here's some Manhattan plots there for those results. So the environmental variable to have the most Snips associated with it was primary productivity with 47 Snips, followed by mean nitrate, which was 35 Snips, and mean temperature with 26 Snips. And mean dissolved oxygen had no Snips associated with it. So yeah. So once we have the genome into the 21 chromosomes and whatnot, we know where genes are, we'll be able to dig in to have a look at where the Snips are sitting. And yeah, it's looking promising so far, but yeah, still quite a bit of work to do. Okay, so to wrap things up, through this project, we were able to create the first genomic resource for Hoki, and the experience of our team meant we were able to progress things quite quickly. And yeah, it was great. The resources, this resource means that, you know, we can dig into lots of other interesting things, carry out other really cool studies, and yeah, especially into things like adaptation to climate change. Although we didn't find any substructuring within the New Zealand samples, we still found that there's a mismatch between management units and the actual genetic units, and the project's been able to produce valuable knowledge that can be incorporated into the fisheries models in the future. Yeah, we developed strong relationships with iwi and industry, and we had a lot of help right across the board. Everyone was really supportive, and we're working quite hard to maintain these relationships moving forward. And yeah, finally, yeah, there's need for holistic approach to management, incorporating all these tools, traditional and genomic and ecosystem-based tools, and yeah, and the more tools we can use to gain insights, it's better for the species, and yeah, more knowledge the better. And that brings me to my acknowledgements. So obviously, Te Ohu Kaimana and Kaiteki, Seafood Innovations and the Deepwater Group, our awesome team, the vessel owners, managers, crew, and MPI fisheries observers for helping with the sample collection, Sheridan Waitai and Doug Jones for cultural guidance, and Peter Van Es and Jeremy Maloka from Neogen for their guidance. And that's me. Thank you for listening.
Thank you so much again, Emily, for your time and all this incredible amount of work that you've presented, and you know, fabulous to have part of the story published and feeding into hopefully future management decisions as well, but also some really exciting things to come. So yeah, in the chat is all sorts of cool Genomics Aotearoa notices, including the fact that Suk Yung will be a research associate professor at Washington State University, will be giving a GA seminar in 2 weeks' time. I'm sure many of us are on the Genomics Aotearoa mailing list already, but Jess has just put out a link there as well to be able to subscribe if you're not on the list. And yeah, thank you again to Emily. It's just awesome to have a Friday afternoon talking genomics. It's been fabulous.
Thank you.
So yeah, noho ora mai. Have a wonderful, safe weekend, everyone.
Cheers.